finally go into the actual pathway, that is the RAS, RAF, MEK, ERK pathway. We have discussed what is RAS. Now we need to discuss what will exactly happen. So we will start from the beginning. We have a receptor tyrosine kinase. It can be a FGFR. It can be epidermal growth factor receptor. Now, if you remember epidermal growth factor receptors, we have EGFR1, then we have EGFR2, and they're often amplified in certain conditions like lung carcinomas, and EGFR2, also known as HER2, is often implicated in breast cancer. The ligand binds to the receptor tyrosine kinase. As soon as the ligand binds, there is receptor dimerization, and that will lead to activation of the tyrosine kinase domains. They will get phosphorylated, they will get converted into phosphotyrosine residues. Now, which was that domain that actually binds to phosphotyrosine residues? Yes, you're right, that is the Src homology 2 domain. The next thing that happens after the receptor tyrosine kinase is that we have a protein known as GRB2. It stands for growth factor receptor binding protein 2. Now, this GRB2 is just a bridging protein. It doesn't have any catalytic activity of its own, it's useless. But it has some crucial domains. That is, it has two SH2 domains and it has an SH3 domains. Now, if you remember, we have just studied this SH2 domain will bind to the phosphotyrosine residues. The GRB2 associates with the receptor. Now, this GRB2 has an SH3 domain, that is a domain that senses proline-rich regions. Now, next in line, we have the SOS protein. The SOS protein has these proline-rich regions. It will bind to GRB2. Now, as we studied, what is the SOS protein? It is a GEF, that is a guanine nucleotide exchange factor. What it will do is it will lead to replacement of GDP with GTP. That is, it is a GEF, and the RAS protein gets activated. Now, this RAS protein, as we studied, is linked to the plasma membrane, and it will basically attract all of the proteins to the plasma membrane. So every secondary messenger system in the initial stages is going near to the plasma membrane. As soon as this RAS gets activated, that is, and GTP is associated with it, its affinity for another protein known as RAF, it increases threefold. Now, what exactly is RAF? This RAF is nothing but a serine threonine kinase. That is, it phosphorylates serine and threonine kinase when it is activated. However, it is not activated. This RAF protein is a cytoplasmic protein. As soon as RAS get activated, it is attracted towards the RAS, but there seems to be a problem, as RAF protein often has excitatory phosphorylation sites and it also has inhibitory phosphorylation sites. And coincidentally, the inhibitory phosphorylation sites are phosphorylated. So we have these phosphoserine residues, and with it we have some inhibitory proteins that have the 14-3-3 domain that often binds to phosphoserine residues, and this leads to its inactivation. However, as soon as it associates with RAS, what happens? RAS will actually activate a protein phosphatase A2, and that will cleave these inhibitory phosphate. We can modulate the activity of kinases as well as phosphorylases with a simple addition of a phosphate group. Now we need these excitatory phosphate residues. Now, how will that come about? There is a protein known as Src. Now, what is this Src? This Src protein is a non-receptor tyrosine kinase. That is, it's not associated with any receptor, but it is able to phosphorylate tyrosine residues. Now, it will phosphorylate these tyrosine residues, and this RAF gets activated, that it is able to do its job of being a serine threonine kinase. Now, as soon as this RAF gets activated, this RAF protein will phosphorylate serine and threonine residues into the next in line in our signaling. That is, if we have covered RAS, then we have covered RAF, then the next obviously would be MEK. Now, this protein MEK, it stands for MAPK ERK kinase, that is MEK, M-E-K, MEK. Now, this MEK is also known as MAPKK, which we will discuss in a few moments. Now, when this MEK gets phosphorylated at these serine and threonine residues, it gets active. Now, this MEK is a very special protein. Why so? Because it is a serine threonine kinase, but as well as a tyrosine kinase. So it actually phosphorylates these three residues. We have already studied we have studied some proteins that are tyrosine kinases. We have studied proteins which are serine and threonine kinases like RAF, but we have never studied a protein which is actually phosphorylating the three of residues. So this MEK, when it gets activated, when it gets phosphorylated and activated, it will phosphorylate the other messenger in this pathway, that is MAPK or ERK. Now, this MAPK stands for mitogen activator protein kinase, or it is also known as ERK, that is extracellular regulated kinase. Now, if you look closely, anything that phosphorylates MAPK will be called a MAPK, that is MAPK kinase. And anything that phosphorylates a MAPKK will be known as a MAPKK, that is a MAPK kinase. So this is known as MAPK, this is MEK is also known as MAPKK, and RAF is also known as MAPKKK. What will exactly happen then? Now this MAPK or ERK, it has various jobs. It can phosphorylate a transcription factor known as ELK1. When this transcription factor, that is ELK1, gets phosphorylated, it can do two jobs. It can lead to the transcription of and ultimately translation of these transcription factors, that is CFOS and CJUN. They often form a dimer, and the dimer is known as adapter protein 1. Or it can actually, ELK1 can also lead to increase in protein synthesis via a eukaryotic initiation factor 4E production that will lead to activation of mRNA. This MAPK or ERK can also phosphorylate CMEK. Now CMEK, when phosphorylated, it is already an active transcription factor. Now what will that lead to? As we studied in the cell cycle videos, CMEK is often associated with upregulating cyclin D2. Now what will cyclin D2 do? Cyclin D2 will convert the unphosphorylated RB into hypophosphorylated RB so that it is acted upon by CDK2 and cyclin E and gets converted into hyperphosphorylated RB. Now, for understanding this, I would like you to refer to our previous videos in which we cover in detail the regulation of the cell cycle. Cyclin D, what will that lead to? It will ultimately lead to the GH transition. That means the cell will proliferate. That is one job. The second thing that transduces is stemness, cancer cell stemness. So CMEK is often involved in reprogramming of cells. That is, it tells the somatic cells that you have to get converted, that you have to go back, you have to evolve backwards and form pluripotent stem cells. Now that is the job of CMEK. Another effect that it transduces is the Warburg effect. Now what is the Warburg effect? We will study this when we will see the molecular hallmarks of neoplastic cells. The Warburg effect is basically anaerobic glycolysis, so that there are intermediates formed which can get shunted into the synthesis of pyrimidines, purines, and the like, so that DNA synthesis can occur at an accelerated rate. It is often seen in neoplastic cells. That sums it up. That is your RAS, RAF, MEK, ERK pathway. A quick revision: we have the receptor tyrosine kinase that will activate. We have some bridging proteins, that is the GRB2. Sometimes we also have other bridging proteins like SHC, SHEC. It is often pronounced SHEC. So we have other proteins as well. Now GRB2 associates with SOS. SOS is a JEF that leads to the replacement of GDP with GTP that leads to activation of RAS. Now RAS activates RAF by activating protein phosphatase A2, which will cleave these inhibitory phosphorylation sites, and the SERC, that is the SERC protein, will also lead to phosphorylation at excitatory phosphorylation sites. The RAF gets activated. RAF will phosphorylate, as it being a serine threonine kinase, it will phosphorylate MEK. The MEK gets activated, it will phosphorylate serine, threonine, as well as tyrosine residues at MAPK. MAPK can function and it can phosphorylate ELK1. ELK1 can further lead to the increase in synthesis of CFOS and CJUN, which are transcription factors which occur often as a dimer. And also it can phosphorylate CMEK that will lead to upregulation of cyclin D2, cancer cell stemness, and even lead to the Warburg effect.